Good morning. My name is Sabrina Geraci-Yee, and today I'm going to share with you some highlights from our new publication and tell you the story of how an old pathogen, Quahog Parasite Unknown, or QPX, got its new name. For those unfamiliar with QPX, it is an opportunistic or facultative pathogen of the hard clam. Here we have QPX seen in histology, consisting of vegetative cells in hard clam tissue. In heavily infected clams, QPX creates nodules, and here we have a very large nodule from which QPX was isolated in 2019. And we study QPX by culturing. The culture inoculum of QPX of a new culture can be seen as the cloudy material, as QPX uniquely produces gel-like mucus. QPX was first described about 60 years ago in Canada after a mass mortality event. But it was not until 1989 that the term QPX was coined and was recognized as the causative agent of disease. QPX was then isolated in 1998 and could be purchased from the American Type Culture Collection, or ATCC. In 2000, QPX was determined to be a member of the Labyrinthomycetes, specifically the Geraci-Yee. In 2001, cohabitation experiments resulted in natural transmission of disease, but the mechanism still remains unknown. In 2002, in situ hybridization experiments confirmed QPX DNA sequences corresponding to the parasite observed in clam tissue. And later in 2007, reinfection experiments via injection of cultured QPX resulted in QPX disease. All of this research fulfilled conscious postulates and clearly indicated that QPX was the cause of disease in hard clams. But QPX still lacked a formal taxonomic name and species description. Why? Well, this was due to conflicting reports of QPX cell biology and an incomplete understanding of its life cycle. The first controversy is about the typical zoospore stage of Geraci-Yee. Developing flagella were observed in TEM, and zoospore-like cells were observed when cultured in sterile seawater. However, attempts to replicate those findings were unsuccessful, leading to the conclusion of contamination. The second controversy is about the bothrosome, which is a unique organelle of Labyrinthomycetes that produces ectoplasmic nets, which have a variety of functions for different Labyrinthomycete groups, such as substrate adherence, motility, and osmoheterotrophic nutrition. Again, 2 studies found evidence of a bothrosome or net-like projections, but others were unable to replicate the findings. The lack of a bothrosome is thought to be related to the abundant mucus production of QPX, which is unique. During some of my early work staining QPX, the culture had to be demucused in order to stain the cells. Here are some examples of the stains we used, which are also included in the publication. We used Nile red to look at lipids, acroflavin, which is used as a method to detect Labyrinthomycetes in the environment, propidium iodide, and DAPI to examine multinucleated stages. Now, the demucusing process is long and tedious, which involves diluting the culture with seawater, passing it through a needle very slowly, spinning down the cells, removing some of the mucus, and repeating this process at least 5 times, sometimes 10 times, until all the mucus is removed and the culture can be easily pipetted. One day I was feeling a bit lazy and had some demucused cells stored in artificial seawater that were several days old. But instead of demucusing a new culture, I decided to check these cells out. And when I looked under the microscope, I noticed motile swimming cells, very similar to the zoospores of other Geraci-Yee, but smaller. This led us to testing the hypothesis that demucused cells incubated in artificial seawater would zoosporulate. These preliminary results were found by accident and may be one of the few times that being a little lazy in science actually paid off. We selected several strains to examine, which were isolated from different areas, some from New York, Massachusetts, and Virginia. Some have been in culture for almost 20 years, while some were newly isolated in 2019 to ensure that culture age did not impact the experimental results. We also selected a holotype or type strain, which is available for purchase from ATCC. Because past observations of zoospores were thought to be due to contamination, the first step was to ensure that our cultures were QPX and contaminant-free. DNA was extracted from the cultures and molecular analyses were conducted using PCR and sequencing of the products. In addition, culture material was plated on marine agar to determine if bacteria were present. All of our cultures passed this quality control testing, including the testing performed by ATCC on our type strain. We then confirmed the phylogeny of our QPX type strain, consistent with previous studies. We followed the International Code of Nomenclature for algae, fungi, and plants. QPX belongs to the phylum the Straminopiles, of the class the Labyrinthomycetes, of the family the Thoracochytrids. Through many experiments and time-lapse microscopy, we were able to better understand the life cycle of QPX, which was much more complex than we had anticipated. Here is the depiction of the QPX life cycle, illustrated by one of our amazing undergraduate artists, who was able to capture each stage based on the microphotographs we provided. Probably the most important discovery is the production of zoospores, which were only produced in a completely demucused culture, meaning that cultures that were lightly demucused, only going through that process 1 to 3 times, would not produce zoospores. In addition, we observed newly settled zoospores would sometimes divide before resuming typical cell growth into a thallus or vegetative cell. And we thought this was interesting because no other Labyrinthomycetes have been reported to do this. Zoospores had typical Straminopilian morphology with 2 flagella, although they were smaller compared to typical Thoracochytrid zoospores. Interestingly, zoospores did not demonstrate attraction to pine pollen, clam tissue, or sand. And here's a great video showing the tiny QPX zoospores swimming around. Jumping into the life cycle, the sporangium type 1 pathway follows the typical reproduction process seen in all culture types, including hard clam tissue. We refer to this pathway as bursting, as the cell wall is retained and spores are formed internally. Under standard culturing conditions, this process is known as endosporulation and produces non-motile endospores. When the culture is demucused, both endospores and zoospores are produced simultaneously from the same mother sporangium. This pathway is similar to typical zoosporulation seen in other Thoracochytrids. The second sporangial pathway is a little more complex. This pathway we refer to as budding, since the cell wall dissipates. This was only observed after demucusing and can produce both zoospores and non-motile spores formed externally, which is why we don't refer to them as endospores. It also appears to be size dependent, with large cells following the top pathway and smaller cells undergoing successive division. Many Thoracochytrids have 2 sporangial pathways, and this pathway is similar to some other genera such as Botryochitrium, Parietichytrium, and Monorhizochytrium. Here's a great video that shows both pathways occurring simultaneously in a demucused culture incubated in sterile seawater. This video represents 7.6 hours in real time, sped up 514 times at a magnification of 200x. We also found a cyst stage with 2 pathways. When nutrients were exhausted in older cultures, usually more than 2 weeks old, some cells would become encysted with either unstratified or stratified cell walls. When nutrients were replenished, the encysted cell would release the protoplasm and round out and resume thallus growth, or immediately divide into new immature thalli, resuming typical growth. Since we did not observe movement of the protoplast, we are not sure if there is also an amoeboid stage, which is common in Thoracochytrids. After we completed the life cycle and confirmed the production of zoospores, we felt it was time QPX deserved a taxonomic name and formal species description. The new taxonomic name for QPX is Mucochytrium quahogii. We created the genus Mucochytrium in recognition of the unique mucus production as muco, and chytrium for the type nomenclature used for Thoracochytrid genera, going along with names that I previously mentioned, in which they all end in chytrium. And then we Latinized the Native American term for hard clam, quahog, giving us the species name quahogii. And lastly, we provide a formal genus and species description according to ICN nomenclature. We have confirmed the controversial zoospore stage, although this definitely raises new questions as to why zoosporulation is triggered by demucusing and how it relates to disease. There was no evidence of an ectoplasmic net observed during any stage of the observed life cycle, leaving the Bothrosome and EN controversy open, as well as the relationship of the production of mucus to a typical labyrinthomycete EN unknown. We have provided a formal taxonomic name and species description, highlighting its unique characteristics, such as the production of mucus and the production of both motile and non-motile spores. The life cycle is much more complex than previously thought, although similar to other thoracichytrids, consisting of two sporangial pathways, motile zoospores, cyst, and protoplast stages. And these stages open up new avenues into understanding QPX disease transmission and pathogenesis, and how these new life stages influence QPX's role as a parasite of the hard clam. I want to thank all of my co-authors, especially my co-first author, Christopher Brionic, and I invite you all to check out our new article, which is available open access, and to view all the beautiful time-lapse videos. Thank you.